Okay, so I'm starting with chemistry and cooking. And you said you would like to know something about meat and about processes during cooking. So first of all, we should think, what kind of things do we have in our food? And the bulk material of our food consists of three things. This is proteins, carbohydrates, and fat. So this is the bulk, and everything are micronutrients. And actually, when we talk about cooking processes, it's mainly these three. We can be with the fat, we can be relatively fast. Fat is a quite stable molecule. We use it for frying, and we actually try not to decompose the fat. We try not to overheat it. And therefore, if we think, for instance, if you make French fries, you try definitely not to burn your fat, and that means the fat is not doing much. It's a lubricant, it is a heat transfer agent in the pan, but otherwise the fat is, and it helps to the taste, but we're not really chemically changing it. But what we chemically change is are the proteins and the carbohydrates. And this is on this basic slide here. So when we cook meat, we actually make chemical reactions there. I will not show you all the things there because we don't actually know it. We know that we break the proteins down and we create flavor molecules like here shown around this meat piece, that we make new chemicals. But the real mix of all of the flavors which we have in food, these are usually hundreds of compounds. We can use gas chromatography to analyze it, but okay, the structure doesn't tell you much. And on top of it, when you have all these hundreds of compounds mixed together, the sensation it makes to us when we eat it is actually depending also on the concentration of the individual chemicals. So if you take one of them alone, you can tell nothing about it. It's really the mixture of all of it and in the right composition, which is really what tastes good. And to analyze this will take many years to go. But we know that we break down substances and create new molecules. And here is the cycle for sugar. We know, for instance, this one here, this is called diketone, and this makes the butter flavor. So when you break down sugar molecules in the heat, you cannot melt normal sugar without decomposition. It gets brown and it decomposes. And this, for instance, is a very strong butter flavor. And you have different ones, fruity aromas and nutty aromas, which make together the taste of caramel. So this is just a sort of introduction. And then we can also ask ourselves, why do we cook our food? Why don't we eat it raw? And there's many things to be said about it. And definitely what has been researched was that when we cook the food, it becomes easier to digest. And that means we need less time for chewing. Our body can absorb the nutrients more efficiently, and therefore it's more rewarding to eat it. And of course, all of these flavor molecules which are produced during cooking, they are sensed by part of it by the tongue, but most of it goes actually through the nose. And there's a feedback cycle in our body. So if you eat something which has calories, somehow this nutrition which we are getting sends signals to the brain. And if this is all good for us, then our brain knows, okay, what smells like this is good for us to eat, and we will create an appetite for it. This is why we love the food which we be fed from our childhood by our mothers, because we are used to it and because our body has learned what is good for us and what's not. And we have one particular taste which is very revolting to us, and it's bitter taste. So a little bit bitter we can tolerate, but very bitter, you are not able to swallow it. You would rather vomit than swallowing it. And interesting enough, many substances in nature which you can find which are bitter are poisonous. So this is not 100% warning, but if, let's say, you would be thrown out in the jungle and you are hungry and you don't know what to eat and you have nothing else to go by, if it tastes bitter, don't touch it. It's probably something poisonous. And if it tastes sweet, it's probably nutritious. And this is how we work. We love sweet, right? And we love many other things. So let's go to the meat. So you said you would like to know about cooking of meat. So how did it start that people cook meat? I mean, we were hunters and gatherers at some point. And at some point, we must have learned how cooked meat tastes and how we can only guess. I could, for instance, imagine there are animals which are burned in a forest fire. There's a forest fire maybe caused by lightning, and some animals die. And the hunters and gatherers are hungry, and they find a cooked animal in the wood. Okay, they try it. They might have eaten raw food, but now they tried this half-burned animal, and they find it's good too, or maybe even better. Maybe this is how it started. And I think about 30,000 years ago, humans were able to make their own fire, and then they could maybe try to also cook their own meat. We can only guess how this has started, but once it was started, it was a big success. So here, as we have our famous mangal or barbecue, and everyone who eats meat usually loves that kind of smell. I mean, of course, if you're vegan, you might find this revolting. This is actually why I asked you. I didn't want that this is only vegans and I start talking for half an hour about meat cooking, but you said meat, so good. Some people really love it and they pride themselves in eating nothing else. Here's a joke about that. So this guy served a salad in a restaurant and he says, "Here's been a mistake. You've accidentally given me the food my food eats." And here he said, if I need 5 courses for dinner and each course will be steak. Okay, so now if we want to cook steak, steak is maybe the hardest meat to cook, beef. It's the easiest to ruin and probably the hardest to make it right. So I will talk a lot about beef. Of course, we have other meats. We have lamb, we have chicken, we have fish. So what I say is partially also for other meats, but partially is mainly it's for beef, because beef is the meat which gets the toughest if you cook it wrong. For instance, if you would cook pork, many of you don't eat that, but pork is easier to cook. It's not so tough and it is a bit more fatty, and it's you put it in the pan and it's probably okay. With lamb, I'm not so experienced. I think lamb is also a bit tricky to make. And chicken, we should definitely always cook thoroughly, but beef, we should actually avoid to cook it all the way through. This is one of the first things how to ruin beef, is to cook it too much. Okay, so let's look first of all what's in the meat. So here, this is a meat piece. This is an entrecôte steak. And so what do you think is the main ingredient in that one?
It looks very fatty.
Mm-hmm. So if we go by percentage, what is the constituent which has the highest percentage?
It should be protein, right?
We hope so.
Fat?
Actually, fat is also, but you don't see it. Actually, the most of it is water.
Yeah.
This is the same in all our food. So this meat here is 65 to 75% of water. Also, our body is like about this composition with water. So most of it is water, but this is not so important for cooking. For cooking, important is protein and fat, as you said. Yeah. But we have to think of the water as well, because the water is making the meat juicy. If there were no water, you couldn't eat it. Okay, so then we have about 20% of protein, which is tissue. It's a muscle, the meat, and it has long muscle cells. These are fibers, which are, of course, in the animal are used to make it move. So these muscle fibers can contract and relax, and with that one we can move. These fibers, these are packets, we will see that. They're surrounded by connective tissue, which is called collagen. And this collagen is very important because it holds everything together. And when we cook, collagen is also a protein, but different from the muscle fibers. We will see what kind of structures they have. So the collagen is holding everything together, and when we cook it, this collagen dissolves. So for instance, if you cook a broth, a chicken broth or beef broth or whatever, and if you cool it afterwards down, sometimes it solidifies. Maybe you have seen that. And this is because the collagen dissolves and turns into gelatin, and the gelatin makes a structure, and if you cool it down, it gets solid. So this comes also out of the bones and also from the connective tissue. And 3% is fat. Now this differs on what kind of piece of meat you have. It can be more or less fatty. And definitely if you want to make a steak, there should be some fat, because if there's no fat at all, it will be very hard to make a good meat, a good steak. We have a very good meat, which is called bone filet. Bone filet has not so much fat, but it is very tender. So that is also, but again, also the bone filet, as I said, the most easy way to ruin a beef is to put it in a pan and cook it all the way through. Then you can be sure you have kind of destroyed it. Okay, so the fat is in between the muscle fibers, and the fat is also connective tissue. And it's of energy storage in the muscles. And also we have some sugar in the meat, because, you know, when we eat, our blood transports glucose, and glucose goes as a nutrient into the muscle and is also metabolized there. So also some sugar is there, not much, but there is. So I can show you, I use as sources, I use internet pages, but also two very nice books. This one here is by Harold McGee, and it's called On Food and Cooking. So this is not a cooking book. It talks about the food ingredients. For instance, it tells you how many % fat is in cow milk, and how much is in buffalo milk, and how much is in goat milk, and all the ingredients, what kind of proteins are in there, and how they, so this background behind the food is very good here in this book. And also, of course, some things are discussed which are important for cooking, but it's not a book with recipes. And the second one, which I like very much, this comes, there's also on the internet, and there's a TV show, it's called America's Test Kitchen. And this is a recipe book, and they make scientific tests on the recipes. For instance, I have seen one where they said, okay, we are always learning when we make French fries, we should put them in hot fat. They said, what happens if we put them in cold fat? And they did that, and they fried them until they were done, and they weighed the potatoes before and after, and they weighed the fat, and they checked how much fat is sucked up and so on. And it turned out the potatoes are less fatty when they fried in cold fat, because not so much water evaporates, and then they don't suck up as much fat. And I'm doing it nowadays like that. I put them in cold oil and heat it up slowly together, and I find them very good this way. So this is what they do. They make soufflés and they bake and everything. And every time they say why this recipe works, and explain also the science behind it. It's very nice. So these are sources I'm using, and I'm also referencing the slides, which I show you. So I'm putting down here where it comes from. Okay, so here we have another picture of meat. So we have muscles, which are fiber bundles. So these are the muscle fibers, and then we have connective tissue around it. And these are these packages of bundles. And in here again, you have this myofibril, and you have a connective tissue around it, and so on. The color of the meat comes from the myoglobin that makes it red, and it depends on how this myoglobin, how much oxygen it has taken in, whether the meat looks grayish or reddish. So if the meat looks a bit gray, it doesn't mean that it's ruined. That might be simply the oxygen content in the myoglobin has changed. So all of this is protein. And then we have also some blood vessels, okay? So now if you look at what happens, so the muscles, of course, we know that they are there for our motor skills. And we have this actin and myosin, they can slide over each other. And this is how the muscle contracts. So this is very important for us, because when we fry meat, our muscle fibers contracting in the frying pan. So the fibers contract and shrink, and that's very important, because while they're doing this, they squeeze the juices out. So we have to be careful. If we overheat our meat, it will simply get dry, not surprising. So let's look first of all, what are proteins? So now we go a bit to the chemistry. So we have here a so-called amino acid with all the atoms here drawn. So we have carbon atoms, 2 of them. We have nitrogen and we have hydrogen atoms. And this R here is a residue, and this can change depending on which amino acid we have. There can be all kinds of things here. Now, this is a simple amino acid. The acid is because of this part of the molecule. This is carbon, 2 oxygens and a hydrogen. Every chemist knows immediately this is the functional group, which we call carboxylic acid. And this molecule, this part of the molecule tends to give this hydrogen into aqueous solution. So if you cook with it, or if you put this just in water, in an acid, we have a tendency of this hydrogen to come off and increase with that one. So this comes off as a hydrogen without an electron, which is a proton. And acid means that the proton in a solution, the proton concentration has increased.
Teacher, may I ask something?
Yes.
I just was wondered, the other side was NH2 and is the base group, yes. And the other side is but still I call it amino acid. Why is it like that?
Amine comes from the amino group. So the nitrogen with 2 hydrogen is the amino group, which gives the name amino acid. So, okay, you could say it's you could also call it an amino base if you wanted to. It's both. It's acid and base. It has 2 ends, no? So I'm not aware where the name exactly was invented. Actually, they have also a structure where this hydrogen moves to the base, as you saw correctly, because this is a base, because it can accept protons from the solution. This is what bases do. So acids give protons to the solution, bases accept them.
So they're actually neutral, not base and not acid.
And then it's a so-called Zwitter ion, because this will become positive and this negative. Then you have a Zwitter ion. This is also a form in which they can be. Okay, but they have these 2 functional groups for the chemist, which is an acid and a base in the same molecule. And we know from our acid-base strengths that in fact this acid and this base have very similar strengths. So if you would put it together, you would probably get a neutral solution in the end, because they are kind of, this cancels each other out. If you give this proton to the solution, this base will accept it. Okay, now when we put 2 of them together, they can react. So we put a second one. And they have an interesting way of reacting. Now you might think, okay, we have acid-base chemistry, so this one should give a proton to the base, but something else happens here. And this is it's not a simple chemical reaction. This happens with the help of enzymes. And enzymes can do all kinds of things, okay? So what you get here is that this reacts with one another. And this might be now for the chemist pretty odd, because you react with the proton from the base and with the OH group from the acid, which is kind of counterintuitive, because then this would act like a base and this one like an acid. But you make here a water molecule, and this is an enzymatic reaction. And so you split off a molecule of water, and what you get is the so-called peptide bond. So this you see when we remove this OH group, what's left here is a CO group, and this is sitting here. And we form a connection. This water molecule comes off. Water is a very stable molecule and will just be removed. And then we will connect this nitrogen with this carbon. And then what's left is NH and OH. And this is what you see here. Here's the, CO. CO is here, and here's NH. And this special unit here, OCNH, is called a peptide bond. So now, of course, if you have such a peptide bond formed and you put another amino acid, you can continue this. You now can do exactly the same thing another time. And this chain will grow longer and longer. So if we make it with 2 amino acids, we call this a dipeptide, because there are 2 amino acids connected and we have a dipeptide. When we make the chain longer, we call it polypeptide. And when we make it even longer, we call it protein. Okay, so their proteins are chains of amino acids. And in nature, we have, depending on this residue here, we have 21 different amino acids which occur in animals and plants. And they can occur in all kinds of combinations in the protein. So we have a huge variability of proteins. But this is the idea of a primary structure of a protein is simply telling us which kind of amino acids are connected to form this protein. Okay, so now we make a shortcut. If you look at it, what we have NH groups, CRH groups, CO groups, NH groups, and so on. And because these molecules are really huge, they have in the end tens of thousands of atoms. It's very difficult to draw it or to picture it. So now we make shortcuts. You see that the RCH groups are now drawn as blue balls, the CO groups as orange ones, the NH groups are white. And then you can form this like a chain of spheres in different colors, which are representing these groups. In this way, it becomes easier. And now this is a helical structure, and this is a so-called alpha helix. Most proteins or many proteins form in alpha helices. Some of them do something else, which is called a beta sheet. But this is the main secondary structure of proteins. So we call the sequence of amino acids, which we have seen here, this is the primary structure. And then how they roll up in alpha helix or beta sheets, or there's another option for the collagen, which we'll see. This is called the so-called alpha helix. This is the secondary structure. Why do I emphasize this? If you compare these connections here along the primary structure, these are strong chemical bonds. It's relatively difficult to destroy them by cooking. But these weak bonds here, which holding this in this helical shape, they are very weak bonds. These are hydrogen bridges. They are at least 10 times weaker than the bonds among these molecules here. And this one we will destroy when we cook very fast. For instance, when you boil an egg, this is what you destroy. So you have a liquid proteins, liquid egg yolk and egg white. And when you put it in water at around 65 degrees, this structure gets destroyed because these bonds are weak. And then you form a network of this thing, and then it becomes solid because it makes a kind of a network throughout. So proteins react in that way, but this is not the end of it, the secondary structure. Now, this whole alpha helix, which is shown here, different colors here, curls up in yet another three-dimensional shape. And this is the tertiary structure of proteins. This is also very labile. So this is very heat sensitive, and usually around 30, 40 degrees, you're already destroying it. This is why enzymes cannot tolerate any heat. If you go with enzymes, let's say 40, 50, maybe 60 degrees, some of them, they will be unfolded and then they function no longer. And enzymes are folded up proteins, and they have very complex tasks in our body. And when you change their folding, they immediately stop working. And then if you can combine sometimes, not for all proteins, but some proteins are actually then clusters of different folded up proteins. This is the quaternary structure of a protein. This one here is hemoglobin, just for an example. This is the tertiary, this is the quaternary structure, and this one here is actually hemoglobin, which is responsible for, in our muscles, to accept the oxygen molecules. So this is what we have to keep in mind when we talk about proteins, that they have a relatively strong connected primary structure, which is sustained very well in cooking, but all of this folding up is very easily to destroy at pretty low temperature. If you uncurl this, of course, these sticky bonds here, they are sticky, these proteins. Proteins can be made into glue because when you unfold them, they stick to each other in different ways. So they stick together, and then they usually become solid. So this is what the example, when you fry or cook an egg, this is what you observe. It is the unfolding, it's called denaturization of the protein and forming it into a new structure. And then you get usually a solid form. The collagen, which holds the fibrils together, is yet a little bit different. The collagen is having a different amino acid sequence, which is called, it has an alpha helix breaker in it. So it prefers not to curl up like that, but makes three-dimensional, makes a helix of 3 different proteins. They are called, actually, the collagen is a protein containing the proline, glycine, and hydroxyproline. These are 3 different proteins, and they prefer to curl up like this. So this is different, and this so-called triple helix is very good for structure material to hold our muscles together. And so this one we can dissolve also with cooking, as I said. When we cook the collagen, which is present in all of the meat fibers, then this triple helix splitting up into individual protein strands, and then you get gelatin, which makes again a 3-dimensional network. If you cool it down, it solidifies. So in the muscles, we have about 1,000 such triple helices to combine into fibers, and these are the so-called collagen fibers, which give stability to the muscle fibers. This is insoluble in cold water, but upon heating, we can dissolve this. So we get then out of it sticky gelatin, which is important for soups and aspics, if you heard of the name aspic. It become muscle fibers. When you heat them, they become tougher during cooking because they contract in the heat, squeeze the water out, but the collagen, the connective tissue, becomes softer. And then the meat separates into individual bundles. Everyone has seen when you have a well-cooked beef stew, then you can, with a fork, divide it. And this is the muscle fibers still stick together in bundles, but the collagen has been cooked out. And then the whole thing, this can be parted with a fork, even without a knife. If you have young animals, they usually have collagen, but collagen kind which dissolves still very easy. As the animal gets older and works its muscles over the years, then this collagen gets tougher and tougher and more cross-linked and less soluble. This is why when we have young animals, they are much more tender than when you have older animals. So old cows are pretty tough. If you have an old ox or old cow and you try to cook this meat, it's much tougher than the meat of a calf. The fat is important because it separates, its connective tissue. Fat melts also when you are cooking, and then the meat can be separated, and it makes it also more juicy and more creamy on the tongue. So the fat occurs under the skin. It's an insulating layer there. It's in all the body cavities around kidneys, intestine, and heart, depending, of course, of the composition of the body. And it is in the muscles, and then it's called marbling. And a good cook will always pay attention to a steak which is well marbled, to have the meat in small bundles everywhere, separated by little fatty tissue. So it contributes to so-called apparent tenderness because the muscle fibers are still tough, but they are falling apart, the bundles, and then you have this interrupts and weakens the muscle. And because it melts when heated, it lubricates, and then we get a very pleasant feeling when we eat it. So if the meat is too dry, too lean, it's not really the best meat. So when we fry the meat, now comes the problem. When we put the meat in hot oil, then the first thing is it behaves like when you boil water. It heats up to 100 degrees and it does nothing else but evaporating water. And you cannot heat it any hotter than 100 degrees until the water has been evaporated, at least from the surface of the meat. And then the temperature goes higher than 100, and then the browning reaction starts. So when you fry and it starts browning, then you have been at around 150 degrees when the browning reaction is most efficient. And this makes then the flavor molecules, which taste very good. This is why we have to go to this temperature. Otherwise, you simply have cooked or stewed meat, which is not so tasty. But so we have different things happening now at different temperature. In the beginning, before you reach 100 degrees, at 40 and 60 degrees, depending, there are different proteins. These proteins start denaturing, and denaturing is unrolling the secondary and tertiary structure. The color changes and the meat turns from pink to gray. The muscle fibers start contracting and they squeeze out the water. Collagen as well. So as you squeeze out water, of course it gets drier, and on the surface water evaporates. That helps also, but also inside, as it gets warmer, water gets squeezed out like when you squeeze a sponge. At around 70 degrees, the collagen starts to dissolve, and the triple helix splits up into single strands of protein. This is called the gelatin. These are these individual strands. And above 100 degrees, the primary structure may also start breaking apart. And what happens on top of it, the proteins react with the sugar in the muscle fiber. So there's a very special reaction, which is called the Maillard reaction, which happens in particular between protein and sugar. And this creates hundreds of flavors, but also some substances which are created, even cancer-causing agents, but these are small amounts. And when we have eaten it for millions of years, so, but it's also creating some poisonous substance.
Teacher, are these cancer-causing agents like radicals in the meat?
There's all kinds of things. Radicals are also supposed, but this is also a tricky story, because it has been shown if you eat or if you give, this is the vitamin story, which is actually diffusing the radicals. Actually, they have made animal testing, and the animals who got vitamin supplements to decrease the radicals in the body lived shorter, not longer, and aged faster. Interesting enough. This is another story which I cannot talk about today, but there were tests also made on this. But so this can be all kinds of substances, not only radicals, okay? So the Maillard reaction, only very short, it's a very complicated reaction. It occurs in all cooking processes. And this is, if you say, I always said it's my favorite reaction. My sister responded, my reaction is alcoholic fermentation. I said my favorite is Maillard because it makes bread crusts, and I love bread crusts. It makes this kind of thing, this browning, this is the Maillard reaction. It happens in bread, it happens on the steak, everything which is browning is the Maillard reaction. So what it does, so we have protein and carbohydrates, because the carbohydrates are delivered to the muscle for energy, and therefore they are also, when the animal is killed, these carbohydrates are still there. And when we heat to about, the best temperature for it is 154 degrees, then it has its maximum reaction speed, and it reacts the protein. So we have here the peptide bond again. We have the sugar molecule. So this is a glucose molecule. This has this carbon chain of 6 carbons, which can curl up into a ring. And here is the chemical structure of it. Sugars have 6 carbons and they have on every carbon atom. So for the non-chemists, when the chemists draw such a crossing, that means there's a carbon atom. We are just too lazy to draw it because it's a lot of work. You see the carbon atoms here, but to draw the structures is much faster to make tack, 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 you know, go around and don't draw these carbon atoms. So there are carbon atoms here. And each one carries an H and an OH group. If we have time for the sugars, I will show a little bit more about it. But so this is the two things which react in the Maillard reaction together. So we have here the protein, and this is the NH2 group, which is reacting here with a group from the sugar, which is an aldehyde group. For chemists, for other people, don't worry. If a carbon atom carries a hydrogen and an oxygen and nothing, and only one other bond, this is an aldehyde. And this can react with our NH group. And again, the idea is we split up a molecule of water. If you compare this and you count the atoms, we have here nitrogen, 1 hydrogen, 2 hydrogen, 3 hydrogen, a carbon and an oxygen. Here I have carbon and nitrogen, And what's missing is the oxygen and 2 of the hydrogen. So a molecule of water went off and a new bond was formed. And then it can rearrange. This is called a shift base, and this is called the Amadori rearrangement. And this continues. This is only the starting point, and then it starts breaking down in hundreds of molecules, which create the good flavors of the food. Okay.
May I ask a question?
Yeah, sure.
We said that the optimum temperature for Maillard reaction is 154. And we also said above the temperatures 100 Celsius degrees, some cancering agents may occur.
Yes.
And do we say that we are sacrificing our health for eating something more tasty?
Okay, this discussion goes on, especially when you barbecue. No? If the fat drops here into the fire and you burn this fat and it goes back as vapors on the meat. So many people have analyzed, and yes, there are chemicals which cause cancer. But at the other hand, there is, for instance, certain vitamins. It has been shown when you eat at the same time vegetables, they can diffuse this problem. And after all, we are living, right? We have tested this kind of process. This is actually one thing which is very interesting. When people started cooking, they had no idea about that. If this would have created too much poison, they would have died when they tried it for the first time. Apparently,
yes, but it's something in long term, isn't it?
Yeah.
Because
sorry. The answer is very complicated because, the thing is, we would like to test such things, but you can normally, scientific testing means the following. You make 2 groups of people. Let's say you take 10,000 people who get one kind of food and there are 10,000 of the other. So you would say, let's give to 10,000 people steak and to 10,000 others, everything else equal, you don't give fried steak. And then you have to observe this because cancer can develop over decades. You would have to observe these people under equal conditions for 30 or 40 years, which is completely impractical. So that means we cannot test it. And if we don't test it, we don't know it. We know that there are substances which we do in animal tests or cell tests. We know that they can cause cancer, but we do not know all of the complicated detail how our body copes with it, because very often when we stress our body with something, it finds a repair mechanism which is actually good for us. This is the same thing with the free radicals. Yeah? It turns actually out the free radicals create a certain stress which triggers a repair mechanism, and this is why it turned out that we try to remove the free radicals from our bodies. We are doing ourselves no good. But there are so many complicated processes that we simply don't know them all.
Okay, thank you.
But you have heard all this discussion about French fries and what kind, and acrylamide in after frying, and should we only eat cooked food or should we not? Okay, you can steam only your food and try to avoid that, but I cannot guarantee you that you will live longer. Okay, so now we are reaching at the point to say what is now the difficulty with the meat? And we have already seen, we have several processes which are happening at different temperatures. And of course, we have one piece of meat. So the aim, what we want to make, we want to have a tasty piece of meat, which is tender and juicy, evenly cooked, and it has the flavors on the surface from the Maillard reaction. And the problem with that is, to create the flavors, we need 155 degrees. That means we have to, first of all, at least at the surface, evaporate the water from a thin layer of the meat, because otherwise you can't reach this temperature because it will stay at 100 degrees and it will not get hotter until all the water is gone. Second, we would like to make the color gene tender. So for this one, we need 70 degrees. And this we would have to reach throughout the meat, also in the middle. Now, the moisture loss and the fiber firming is actually starting at 55 to 60. That means at this point already the meat starts getting dry and hard. So we need to reach these temperatures without overdoing because otherwise we are losing too much moisture and the meat gets like, you know, what we call a shoe sole, which nobody can eat anymore. So now we have to find the best compromise for that. And we also have to choose a good meat. So I'm showing you another thing. Justus Liebig in 1800 something, he is a famous organic chemist. He made lots of contributions to biology, agricultural chemistry. He organized organic chemistry. He invented the organic chemistry lab. He was one great chemistry teacher and so on. And he was also very much involved in food. There was a lot of poverty at the time, and he was realizing that fertilizer needs nitrogen. And finally, he invented the bouillon cube, because he believed at the time that when you cook meat out, that all the nutrients and the good stuff is in the broth. And then he made a concentrate and he wanted to feed people with that. Okay, so he made a claim. And this is a claim you which have probably heard. Many cookbooks are saying it, many advertising saying it. They say you have to sear the meat at very high temperature. That closes all the pores, and then the juices are staying in the meat. Maybe you have heard that the meat has to be put in very hot oil only in order to keep the juices inside. Okay, we tested. I bought 2 pieces of meat. I'm a chemist. I like to make experiments. 2 pieces of meat, equal weight. I put them in 2 equal pans on the stove and put the flame, one on full heat, one on minimum heat. Fried each one 2 minutes from each side, and then weighed the meat before, 135, they were adjusted. Weigh it afterwards and look for the juices. What's happening when I make it hot and not so hot. Okay, so here's the picture. This was rump steak, not very marbled, but we're not trying here to make the perfect steak. This was the test for which one is losing more juice. Here are the 2 pans. These were the 2 equal ones I had. The flames, low and high. Put a bit of oil, which is the heat conductor. Stopwatch, 2 minutes on each side. So you see here is the hot pan. Clearly it's smoking. This is actually, when you see such a smoke, we are over the temperature what the oil can tolerate. So depending on what oil you use, some oils tolerate more. Olive oil cannot be very hot, but this is probably over already 240 degrees when the oil starts making that. Not the healthiest way of cooking. This one is the cold one. Now, if you look carefully, you see here juices, right? Coming from the cold. On the cold pan here, you see it looks pretty dry. We continue this game. Here, very hot. So now here was still juice. Now it gets a bit less. It's drying up. Now flipping it over. Of course, the one in the hot pan is much more brown than the other one. Doing it 2 minutes and then take it out. Now I weighed the 2 steaks, and the result was the steak from the hot pan had lost more water than the one from the cold pan. The hot fried steak lost 10 grams of mass. This is water which evaporated, and the less hot one lost 7 grams. So why did it look like the juice came out of the cold one? Because in this hot pan, it was evaporating so fast that we couldn't see the liquid. It was simply evaporating faster, and it evaporated more because it was hotter, and the sealing on the surface doesn't work, right? Okay. Then after waiting 10 minutes, here almost no juice came out, and here even more came out. That means, what is the conclusion? Upon cooling, more liquid came out of the hot state than out of the cold one. Liebig was wrong. Proven, right? Without any doubt. So this searing, we still fry it hot because we want to have the browning reaction. But that the juices stay inside is clearly wrong. If you make it hotter, it gets drier. It doesn't help. The reason is the muscle fibers contract when heated and they squeeze out the juices. That will not be the best steak. So if you make it too hot and too long, especially, it gets dry. But this meat was also not finished cooking. This is inside raw. There was, of course, a mistake because what I did, the cooking for the meat for 2 minutes left it not finished. If I would fry it longer, it would get probably quite dry because you see there's a pretty big layer already, which is cooked and it's already getting dry. So then also the meat was too lean, the meat was too fresh, and it was too cold. It was from the fridge. Now let's make it better. Okay, so the first thing what we should do is aging of the meat. So do you know aging? Have you heard about it? I found in Turkey you don't get aged meat. It's always fresh. Problem is, so what happens in aging, that means is the following. The animal is killed and then you know what is rigor mortis. A dead body becomes stiff, right? This is when the muscle fibers start contracting. So if you have a meat slaughtered and then you, after a couple hours already, it will be very hard. Now you have enzymes in this meat, and these enzymes are normally responsible for digesting the food. And they are still active. You kill the animal, but these enzymes are molecules, they continue. And instead of now digesting food, they digest this meat. That sounds maybe disgusting, but not at all, because it makes this meat tender and it creates flavors. The meat starts changing its taste and it changes its structure. So during the aging, the proteins are broken down and you form amino acids. The glycogen, which is a sugar polymer, goes to glucose. Adenine triphosphate is there and inosine monophosphate, and you have lipids and they are broken down into fatty acids. And the whole thing creates flavor. The meat becomes much tastier. In the 19th century, they left the meat for a month at room temperature until it was stinking, and they called it mortification. It was really a fashion to do it. Nowadays, what is done to very good meat, it is kept at 1 to 3 degrees. This prevents growing of bacteria because it's even colder than a refrigerator. And you need high humidity that it doesn't dry out. And then it can hang for a month or longer. And this meat is much, much better than fresh meat. If you buy well-aged meat, you pay a lot more for it in Europe. I don't know whether you can get it in Turkey. Now, in order to make a good meat, we also choose a little bit fattier. We have said, for instance, entrecôte, like it's called here, is a good meat for frying. I left my steaks, which were fresh, I left them now for 5 days in the fridge to make at least some aging. Before frying, I warmed it up, no longer from the fridge directly. I put it actually in a plastic bag and put it in lukewarm water in order to get it at least a bit above room temperature. And then I made 2 kinds of different steaks. One I fried for 2 minutes on each side like before, and the other one I flipped every 20 seconds. This is also a procedure which I found from good cooks in the internet. Some of them swear on it. Okay, so this is the entrecôte fresh, and this is the entrecôte after 5 days. And what you can see is, so this is all compact here, but when you leave it in the fridge, you see already that this is all opening up because the connective tissue, because of the aging, is broken down by the enzymes. Also the color changes. This is not rotten. You have to follow your nose there. This can be probably aged a bit longer even, but it helps already 5 days. And then put it in the pan and do the same cooking procedure. So now this one on one side for 2 minutes, this flipping. And the temperature was very hot on both sides to get the browning reaction. So you see this is flipped, 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 and this is not. And keep going. Now this one is flipped. It's of course fried a bit more because it was all the time on this side, and then keep going. The oil is smoking. I would probably not always make it that hot. And then take it out. So now after doing this, you should let it rest for about 5 to 10 minutes. This is usually also said because if you cut it immediately, it loses too much juices. And then you cut it open and you see this time the meat is cooked. I have to, well, I don't know. You have to let it like that. You see, because it was warmer, this meat is finished. It's cooked. It's not raw anymore like the other one was. And we see that this one, which was from one side for 2 minutes, and then the other is not as well cooked through as this one. But there's not much difference. I ate both of them, and if you had asked me with closed eyes, I would not have been able to tell the difference from the taste. If you compare it to the older one, this was raw inside because it was simply too cold. So definitely never, ever make a steak right from the fridge. And if they tell you you shouldn't leave it on the counter for 2 hours because of bacteria, well, you know, bacteria grow, but you could, as I said, you could use a water bath like I do it, but this is much better. Okay, and then you see that this is how a steak should be. Another option would be, if it's a very thick one, fry it 2 minutes from each side and put it in the oven. I'm sorry, I have to take this for a second. Hello? I'm teaching. I mean, I can quickly open the door, but I cannot talk now, okay? Okay, all right. Sorry for that. Okay, so we are at the end. I just have to quickly open the door. You can have a quick look at this slide, and I'm back in 1 second. Okay, to sum it up, how to cook a decent steak, first of all, choose the right meat. It shouldn't be too lean. And if you cannot buy aged, try to age it yourself in the fridge. Be careful, it should be at low temperature. It shouldn't dry up, but it gets much better if you leave it at least 5 or 6 days. Fresh meat simply doesn't taste good, and it gets hard because all this collagen is broken down by the enzymes, and it helps us. Take it out of the fridge and use it warm. Don't make too cold meat because you cannot fry it evenly. You would overcook it on the outside and you don't get it done on the inside. If the pan, it has to be hot enough to ensure browning. This has nothing to do with sealing in juices or anything. It's important for the taste. You want to get the browning reaction for the flavor, and aim at about 70 degrees inside. That's enough to make the proteins react. And avoid overcooking. Flipping it helps. And if you have a bigger piece of meat, as I said, you can sear it and then put it in the oven at low temperature and let it reach this 70 degrees inside, and you will get a tender and nice meat. So it took longer than I thought. So I think we should stop here, and I will not talk anything about sugar. I think this is enough, otherwise it's getting boring. So this is what I can tell about meat cooking in short, and I'll stop at this. If you want to ask anything or comment anything, just that would be nice.
We can take the questions now. If someone has a question, they can raise their hands. Anyone? Yes, Burhanettin, go on.
I want to talk about the proteins we take to our body, and if it exceeds what we need, what happens to that protein? Okay, it will be metabolized. So the protein is also, our body prefers at first, the first energy source our body is using is sugar. The second one is fat. And if the body does, what it does with the protein before it burns it, it breaks it down into amino acid and builds new proteins. So proteins are a very big building material for the body, because there are proteins which our body can only make from animal amino acids. So we need animal protein in order to reconstruct our own proteins, and some proteins we need to take with the food.
So the body prefers to use the proteins, first of all, as a source to synthesize new proteins. If you eat more, it will be metabolized. So it goes, it will be oxidized. The carbon goes to carbon dioxide, the hydrogen goes to water, and the nitrogen and any phosphorus or sulfur which is in it will be disposed through the kidneys and goes to urea. So in fact, if we eat too much protein, we could actually get problems with crystals in our joints because the kidneys cannot clear it efficiently anymore. So it can be too much and too little.
Could you please share your slides about the sugar? And I'm really curious about the next slides. I don't know.
I can continue if you wish. I mean, I have no problem with it, but I didn't want to bore you because it is 1 hour already. But I can continue, no problem. We can also make, whoever wants to stay can stay, and who doesn't want to doesn't have to. Yeah, I could also, if you...
Time is not a problem for us. If someone wants to leave, they can.
Okay.
Yeah.
So if you have more questions, I can answer. And if not, I can continue if you wish. I mean, no problem.
I think some people want you to continue.
Okay, good. Yeah. All right. Any more questions before that? No? Okay. So sugar, this is carbohydrates. Now, a sugar molecule, you have seen quickly a structure before. But so we have 12 carbons in a sugar. We have hydrogen, 22 of them, and we have 11 oxygens. Now, if you quick in counting, we can write this a little bit different. Why is this called carbohydrate? We can write it like this. So we have still 12 oxygens. We have 11 times 2, 22 hydrogens, and we have 11 oxygens. So we can make it as a carbon molecule with 11 molecules of water. That's the carbohydrate. How can we prove it that this is true? Now, for the chemist, this is a fun experiment. We can pour sulfuric acid on it. Okay, so this is sulfuric acid. You need concentrated sulfuric acid for that. And sulfuric acid has the habit of very strongly binding water. So what it will do when you pour it on sugar, it will take all of these water molecules out of the sugar, and what's left is the carbon. That looks like this. It gets a bit yellowish, and then the fun starts. It gets black, and it starts growing. That's what you're getting from sugar and sulfuric acid. So that's the carbohydrate, okay? What happens is the sulfuric acid simply takes all of the water molecules from our carbohydrate and leaves the coal, the carbon behind. So if we eat it, from this book on food and cooking, the guy said sugar is sensation, pure sensation, crystallized pleasure. Why? Because when we eat sugar, this is the first energy source our body has. You know, in our blood circulates glucose, and the glucose goes directly into the muscles, into the brain. And this gives us energy. And because of this energy, we get a positive feedback from our brain to eat more of it. So this is why we love sugar. So this is the sugar molecule. This is the similar picture. So sugar consists of 6 carbon atoms. This is the glucose, and we have these OH groups. So hydrogen, so carbon has always, it makes 4 bonds. It can be 4 individual bonds, or it can be double bonds. Then it needs 2 others. Again, if you count this as 1, 2, 3, 4, 4 bonds. And this sugar molecule normally is forming a ring. It reacts here and closes this ring, and then it looks like that. So this is the form of glucose. This is the same structures in a so-called ball and stick model, in which we show the atoms as balls. This is the same thing. You see, this is not so easy to spot it here. Actually, in this kind of drawing, it's easier. So this is the first structure of glucose. Now, we can also, you see all these OH groups. In glucose, it's like that. There is, in fact, 16 different ways of arranging these OH groups when you make all permutations and make the mirror images. And one of them, the most stable one, is glucose. This is the one which is very common in nature. You know probably about a second sugar, which is called fructose. Fructose has a slight difference from glucose. If you look here, we have on this carbon 1 hydrogen and 1 oxygen. On this carbon, we have 1 hydrogen and 1 OH group. If we switch this here and put this hydrogen and this hydrogen up here, you get the OH group and the second hydrogen here. Then you get this here, and you have this double bonded oxygen moved down 1. And this is the structure of fructose, a very small modification from the glucose. Now, this can also fold up, and the glucose usually folds up into a ring of 5, in a 5-membered ring, can also fold up in a 6-membered ring, and this is called fructofuranose and fructopyranose. That's the name of these rings. Okay, so the fructose is occurring also in nature, in fruits and so on. And we can combine the two, and this is called sucrose. This is the sugar you have on your table. So it has 1 molecule of glucose and 1 molecule of fructose together. They are connected here. And if you count all the atoms, you get this C12H22O11 structure. So this is what we are eating. Now the sucrose is a so-called disaccharide because it has 2 sugar molecules connected, therefore disaccharide. It contains equal amounts of glucose and fructose, so 50% each makes of sugar. And that occurs, for instance, in pineapples, in apricots, and many things. And we have fructose is the majority in pears and grapes. And fructose is actually sweeter than glucose. I have here the sweetness. So we have different sugars. I didn't show all of them. The milk sugar is called lactose. It's not very sweet. We have a couple of others. Here's glucose. We are all doing this related to sucrose, the table sugar, which has 100%. And then you can see that pure glucose is less sweet than sucrose. Honey, which is more or less sugar solution, nothing else, is 97% of sucrose. And here is what's called high fructose corn syrup. This is what now the food industry is using a lot to sweeten Coca-Cola, Fanta, food, all kinds of things. High fructose corn syrup is very cheap because they can do it from corn. And this is used a lot in industry as a sweetener. And here you see the sweetness of fructose, which is almost twice as sweet as sucrose.
Teacher, does the sweetness change the tendencies of fructose and other sugars to turn into fat? So has it something to do with the sweetness?
It has not to. The sweetness is because the detection on our tongue. The receptors for taste on our tongue. There are different places. These are relatively big molecules, and they touch different receptors in a certain geometric arrangement, and that sends to our brain through the nerve from the face, from the tongue, sends a signal to the brain, says this is sugar. So you have, for instance, some artificial sweeteners which can be 100 times more sweet than sucrose. The metabolism is a different story. Now, I have a little bit about the metabolism, which is coming now, so I think I'm postponing it. But I can say as an answer here, sucrose is first of all burned, led to the muscles and is consumed there. And only when we eat excess of sugar, then we will turn it into fat. Fructose cannot be directly used. It needs always to go to the liver, and it will always turn into fat because our body cannot use it directly.
But is it not same for the other sugars than fructose?
Sucrose not.
Okay.
I will give you a little bit more about it. So here you have a list just for where these occur mainly. So we have here total carbohydrate content of some fruits and vegetables, how much total sugar it is, how much is free fructose, how much is free glucose, how much is sucrose, which is the disaccharide. And then you can see here the percent of sucrose of the total sugar and the glucose fructose ratio. So this depends on the fruits. So if you say, for instance, apples, you look, there's more fructose. In apricot, you have more sucrose and so on. So it differs. Very high content of dried figures, because when you dry fruits, you remove the water, that lowers the weight, and then the percentage goes up. No? So this dried one is actually not a good comparison to fresh fruit. But you see, for instance, peaches are having a very high sucrose content and so on. If you look down for the vegetables have less sugar than most of the fruit, but for instance, sweet pepper and some sweet potatoes also have a lot. We have another species, which is the polysaccharides. You see, this is just units of sugar connected into polymers. And this is what starch is all about. So starch is a polymer of sugar. This is also a carbohydrate. So it's just a very long molecule of many, many sugar units. And what's very interesting, if you compare starch and cellulose we cannot digest. And the only difference is, look here, if you look at these sugars, the oxygen points down, and here the oxygen points up. This is our main source of energy in our body, carbohydrates, and cellulose we cannot digest. Chemically, very little difference, but the difference are the enzymes. Our body has no enzyme to break this down, and therefore this is dietary fiber. When we say we should eat high fiber diet, cellulose is a plant material which we cannot digest. Very similar to starch in the chemical structure, but physiology is very different, because our enzyme which breaks down this one cannot work on that one. Okay, so starch is the main energy storage molecule in plants, and we eat it through flour, through potatoes, through rice, and so on. And we have a little bit different starch molecules. We have a linear form, which is called amylose, and we have a branched version, which is amylopectin. They are arranged in the foods in microscopic granules, and then you know, we have the flours, we have all these things. Polysaccharides contain many OH groups, and they can form hydrogen bonds. They can absorb a lot of water. When we make bread, this is very important. You put water on flour, and if you ever bake the bread, you use about 70% water in bread. So use about 1 kilo of flour and about 650 to 700 grams of water. And this is bound in the starch because of the OH groups which can connect to the water molecules. If we have starchy tissue and we cook it in water, the starch granules also absorb water. They swell, they release starch molecules. This is used for thickening sauces. So we can make our sauce thicker by putting a bit of flour or starch into it. And when we cool it down, this is rebonding, and it forms a kind of a moist and solid gel, which acts as a thickener for our food. So the cellulose is the structural support, which is in the cell walls of our as tiny fibers. And most animals cannot digest it at all. The cows can. Cows and sheep, these are the so-called regurgitating animals, can, but only because they have bacteria in their stomach which can do it. So these regurgitating animals, they chew this food again, it comes back up and they chew it again, and the bacteria breaking the cellulose down. So the sheep and the goats and the cows, they can get energy from cellulose thanks to the bacteria in their stomach. And the cooking leaves the cellulose also intact. So this is for us the dietary fiber, which is indigestible. Now comes the storage. So if we eat sugar, we produce glycogen. Our liver is doing that. We call this animal starch. The glycogen is just a sugar polymer. So what it is like, there's an enzyme and it creates such a branch structure, and every little thing is a sugar molecule. And this is the glycogen. And so when we eat sugar, what happens is, as I said, if we are getting hungry, which is usually a sign that our blood sugar level has decreased. There are some other hormones which work as well, but we have a detection for low blood sugar level, and that usually induces hunger. So we eat sugar, our blood sugar level goes back up, and the normal thing is that our pancreas produces insulin, and the insulin induces our cells to take the sugar in. So if we are trained athletes, our muscle cells are very good in using sugar, and the sugar goes into there and is metabolized. If we eat more sugar than we need, then our liver produces this glycogen. And this glycogen, you can see it's about 30,000 glucose units, such a glycogen cluster. And the liver can store about 120 grams of that. So 120 grams of glycogen goes to our liver. And when the blood sugar level goes down, this all is governed through hormones. And then new hormone says to the liver, please, we need sugar. Liver decomposes the glycogen, sends it back into the bloodstream, and it is again metabolized. If we keep eating sugar and the glycogen storage of about 120 grams is full, then the liver synthesizes fat out of this sugar and puts it in a storage, because fat can store much more energy for the same amount of grams than the sugar, the carbohydrates can. So it's actually breaking up the sugar molecules and makes fat molecules out of it. Then we produce fat. But fructose is different. Fructose cannot be used directly. It goes into the liver and it is put into fat. And then if you want to burn it, you have to, again, starve yourself of all the carbohydrates to induce your body to burn the fat. So fructose is much more fattening than glucose. So if you eat too much fructose, that's a problem. And the other thing is that glucose, because it triggers the hormone insulin, our glucose level are connected with our feeling of hunger. So if we have enough glucose, there's another hormone, I think it's called leptin. It induces us, there's enough sugar there, do not eat more. But the fructose doesn't trigger that. So the fructose provides energy, makes the body to create fat, and it does not produce the feeling of being sat, being full. You are still hungry when you eat fructose. You're producing fat because you can't use it. The body has energy, but doesn't signal to the brain, stop eating. This is the danger of fructose. So they did, in the beginning, they thought our diabetic people should avoid glucose to get their blood sugar level down. And they made diabetics products with fructose, and the people got very fat because this is not good for them. Okay, so the last thing which I have about it is what happens to sugar when we heat it. Then there's what I don't show today, but you can cook sugar with water and you make candy making. This gives you a very solid stuff. But just to see what happens when you try to melt sugar on the stove. So here we are already at 128 degrees. So the sugar starts melting, but watch it. Before it's melting, it's already getting brown. The sugar melts at 186 degrees. So we are here at 159 and it changes color. So before it melts, it starts decomposing. So sugar cannot be melted in a complete structure. You decompose it, it gets hotter. Eventually it starts melting. It's very hot. One has to be careful. This is 170 degrees, much hotter than when you work with water. So it's really, really hot. And then finally we can melt this stuff. Here I have a nice melt here. And this one, once you have melted it completely, I spread it out on a parchment paper. This is caramel. This tastes good. I used it. I just was eating some of it. I made some into the tea. Really, really tasty. So the sugars, when you cook with them, either they react with the proteins and make this Maillard reaction and make flavors from this point. If you cook it alone, it decomposes and creates flavor. And if you cook it with water, this is the candy making process. You can form all kinds of things and structures with it. So sugar is also very interesting for cooking. And the browning is used a lot. This kind of caramel browning is to color industrial food. They use a lot of sugar products in order to change the color of the things and also to flavor it. And this here is really tasty. If you don't make it too dark, this tastes really, really nice. All right, so this is called caramelization. So this is what the sugar molecule, this is the sucrose molecule, and this is what you get out of it. I told you this is the butter flavor, and there are some others. You create all kinds of things there. And this is, I think this furan is a fruity flavor. And all this mixture together makes this caramel. So this is also very nice. You can use it for flavoring things. Of course, when we cook jellies, we put a lot of sugar and we're heating it. So part of the jelly cooking is also breaking down sugar molecules. So just the last slide, why do we cook? Well, number one, it tastes good, right? It tastes wonderful. And this is very important because what tastes good to us is something which we give to our body and it says, yes, give me more, this was good for me. If something is sickening you, you get a feedback from your brain, never eat this again, right? If you eat something which makes you sick, you don't touch it a second time. So our taste is in fact probably through evolution developed over millions of things to warn us from poisonous food like alkaloids in nature, which are usually bitter. It warns us from it wants us to eat rotten food, which is usually tasting sour. And if something is good, we try to get more of it. So the food on a chemical thing, it becomes easier to digest. And probably because of that, again, our body is happy. It gets the nutrients out of it and develops this feedback cycle to say, yes, eat more of this. So when we eat to get energy, our brain responds to this benefit and induces us to seek the source of this food again, which is good for us. Of course, we should do it with moderation, because if we eat too much, obviously, especially sugar and fat and everything, if we eat too much, it's not good for us. If we gain like 150 kilos, this cannot be good. So we need to listen to our body also when it says enough, stop now, please. Good cooks, of course, they know how to create dishes which stimulate all our senses and give us the utmost pleasure. Usually cook says you have to stimulate all the senses for sweet, sour, savory, which is the umami flavor of meat, which is amino acids. And if you make this in the right mixture, the food is just heaven. And understanding these processes behind cooking, I think, is real fun for the chemist to search on that. And with that one, I have actually concluded everything I had prepared for today. All right.
Okay, thank you very much.
Mm-hmm.
We can take the questions again.
So it was enough, apparently. But 52 people were hanging on, I must say.
Yeah.
Bravo for Sunday evening. You're very brave. Did you have dinner already?
I do.
We have warned them, hocam. We said come after eating. You may get hungry.
Mm-hmm. So Elif want to say something. Mm-hmm.
Elif, go ahead.
Yes, thank you, Professor, for the presentation, first of all. I wanted to ask, so when we cook on stove or in hotter places like oven, we kill most of the nutrients, and while we are also killing the bad stuff, we are mostly killing the good stuff also. So what will be the best way to cook in order to get the most nutrients?
Well, I wouldn't say you kill, well, the question is what you call nutrients, you know. You're not killing really the nutrients. The protein is still there, the fat is still there, the carbohydrates are there. They have just, I mean, you have, the molecules have changed, and you're not actually decomposing everything. I mean, you are creating some flavor molecules, but you don't decompose everything, so the calories are there. We have, of course, in our body, very elaborate metabolic cycle, which can make use of many things. No? It can make use of fat, of carbohydrates, of different ones. But you have also seen that like cellulose, we cannot digest, but we're not killing in the oven everything. We do kill a lot of vitamins when we cook. So vitamins are often sensitive molecules. And for instance, vitamin C, if you cook, vitamin C is pretty much gone. So this we need from fresh food. So we should, if you want to eat healthy, you need to eat a good mix of different things. And we should combine, I think there's nothing wrong with meat, but we should combine it also with vegetables. Vegetables are good for us because apparently these cellulose fibers are very good for us. We are used to that. We need micronutrients. I didn't talk about that. Micronutrients are the minerals. We need some sodium. We need some kind of, sodium chloride. We should eat about 5 grams a day. So we have to have not too much salt, not too little. And fresh fruit, vegetables is apparently very good as far as we can test it. It seems to be that the vegetable contain a lot of cancer-fighting agents. This is because if you think of plants, plants are steady organisms which cannot run away from the sun, which is, for instance, shining, making with UV light. It can create breakdown chemicals, and the plants have developed repair mechanisms. And we are eating these molecules which make these repairs, and therefore plants are quite good for us. And for the vitamins, we should eat something also raw food, also salads also. No? Vegetable salad should be definitely in our diet. And in general, it's not good to eat too much industrial processed food. I mean, there's a lot of, it's not really the food additives which are so bad, because they have been tested for safety, but the industrial food, which when you buy finished food, they're putting all kinds of imitations, and this is not good. If you have a lot of starches, for instance, which they process in such a way that they appear to be like meat, but if you look on the package, you have something which has on the side, on the picture, you have all these meat cubes. And when you look how many % of the package is meat, and you find that this is like 7 grams or so, then you ask yourself, what are these cubes which I'm eating? And these cubes, they can imitate meat quite well, but you do it usually with kind of starch-related molecules. So they can nowadays imitate almost everything. And that's not good for us, because when you think of chocolate, you think chocolate. Your pancreas produces insulin. So if you eat now something which is not sugary, let's say artificial sweetness, the pancreas produces insulin because it expects sugar, but it is not sugar. Then your blood sugar level falls, you get hungry, and you eat too much. So all these imitations of food, which are not what we think they are. For instance, you eat a yogurt, which is called strawberry yogurt. It contains half a strawberry, and the rest is artificial strawberry flavor. You think you eat strawberries. You think you eat vitamins, you eat none such a thing, you eat a flavor, and that's it. This fooling of our body is not good. So if you want to eat healthy, I think you should go to the pain and cook yourself. Fresh food, make it yourself, you know what's in it. Eat slowly, don't eat too much, and that's probably the healthiest way. And as I have said, my area needs 150 degrees, so there is probably no need to dangerous substances you create. So if we fire, Yeah.
John,
So
the internet
I would not cook.
not stable.
Yeah, but the oven is not a bad way to cooking, because actually we don't. In my course I was discussing in the oven the heat transmitting is through convection and through radiation, and this is why in the oven you usually cook slower. And slow cooking is not bad. But definitely I would say cook fresh food for yourself. That is better. I mean, it's not poisonous to eat once in a while a prepared meal from the supermarket. But if you eat it only, you eat a lot of things which you have no idea what they are. If you check the package, this is also something interesting to discuss, these food ingredients, what they're putting in there in order to make this packageable. It has to be lasting. If you make a complete meal which you want to put in the supermarket on the shelf, and it has to live at least 2 weeks there, and then it should look good and taste good. You have to put all kinds of things in it to make this possible, because food would otherwise simply be destroyed. And this is a quite unnatural cooking. And it's not poisonous to eat it once in a while, but to eat only this food, I think it cannot work.
Thank you, Professor.
Okay.
Any other questions? By the way, we have a feedback form for every event, so we would like people to fill it.
Yeah, if there are no other questions, Professor, I would like to thank you for coming today. And the topic was really interesting and it was really nice. And I hope everyone feels the same. And I also saw it in the chat that some of the students from the microbiology department wanted to take this course from you, but it was unavailable, as you said. So I hope this was satisfactory for them, at least a bit. I mean, this was like a brief, not a course, but I hope this was nice for everyone. And once again, thank you for coming.
Well, thank you for inviting me. It's fun to be with you. It's fun to have students who are interested in something and to talk to them. And it is really a pleasure to do this. So thank you for your attention on Sunday evening. And I say goodbye and sleep well and see you tomorrow, some of you.
Take care, Jen.
See you, Jen. Take